After the extraction of the DNA, the concentration or the ethanol precipitation is going to be done. Ethanol precipitation का फ़ायदा हमें ये है कि हम DNA को concentrate कर सकते हैं. अगर उसकी जो concentration है that is very poor, we can concentrate the sample so that in future we have to use low volumes, then we can get our required amount of DNA in that particular low volumes. For ethanol precipitation, we need 100% cold ethanol and 2 volumes are going to be added into the DNA sample already extracted from the bacteria or from any microorganism. And after mixing, we have to leave it on ice for 10 minutes. Here we have to look at the size of the DNA or the concentration of the DNA to decide whether it should be stored on ice or under some different conditions. If the size of the DNA is less than 1 kilo base pairs or the concentration is less than 100 nanograms per milliliter, then after ethanol addition, store it at minus 70 degrees centigrade for 4 hours. If the size of the DNA is less than 0.2 kilobases, then an addition of 0.01 molar magnesium chloride will improve the recovery. Once the incubation time is over, we have to centrifuge the DNA sample at 4 degrees centigrade for 10 minutes at 14,000 revolutions per minute, and by this centrifugation all the DNA which is found in the sample will be settled as a white sticky pellet. Discard the supernatant by inverting the tube and remove the traces of the supernatant by using vacuum desiccator. Now the pellet, which is 100% pure DNA, will be dissolved in the 25 microliter of TE buffer at pH 8, and this is going to give us the concentrated DNA sample after resuspension. It is good to rinse the walls of the plastic tube with our buffer so that any DNA which might be on the walls of the tube should also be part of the solution. If the pellet is not successfully dissolved, then heating at 37 degrees centigrade will facilitate the DNA solubility in the TE buffer. Once this solubility is done, then the DNA can be stored at 4 degrees centigrade and further can be used for DNA quantitation.